guys, did you know that over 50 billion of the cells die because of a process known as apoptosis? That's right, it is a vital process. So today we are going to dive deep into the topic. So if you are interested, please stick around. What is apoptosis? Apoptosis is a programmed cell death. Conditions of apoptosis we will see first, and then we will see the process of apoptosis. So apoptosis can be both physiological and pathological. So we will see the physiological examples of apoptosis first during embryogenesis, second after leukocytosis. There is increased WBC which are destroyed by a process known as apoptosis. Third, in immune response, there is suppression of self-reactive T cells. Fourth, chemotherapy. In chemotherapy, there is both apoptosis and necrosis. Fifth, post-menstruation. There is endometrial lining shedding off going on. This is due to the apoptosis. These were the physiological examples. Now we will see the pathological example. Viral hepatitis is a very important example of pathological apoptosis. Apoptosis of hepatocyte is known as Councilman's bodies. So we will see the process. This is an normal cells cut section, and these are the red ones are damaged organelles. Further, the damaged organelles come in one side and the body gets separate as seen in the diagram. So the normal cell gets separate and damaged cell gets separate, and the damaged cell is eaten by the macrophage. This whole process is known as apoptosis. So apoptosis has two main pathways, intrinsic pathway and extrinsic pathway. So we will see it. Intrinsic pathway is also known as mitochondrial pathway, and extrinsic pathway is known as fast ligand pathway. We will see the intrinsic pathway first, and then we will go to the extrinsic pathway. So let's get started. When cell injury occurs, it is sensed by apoptotic sensors. What are apoptotic centers? These are basically proteins. There are four types of proteins: BH1, BH2, BH3 and BH4. But in intrinsic pathway, BH3 proteins are involved. Examples of BH3 proteins are BIM, BID, BAD, NOXA and PUMA. It is in mitochondria. In mitochondria, there are two types of genes: pro-apoptotic genes and anti-apoptotic genes. Pro-apoptotic genes increases the process of apoptosis, while anti-apoptotic genes decreases the process of apoptosis. Therefore, it is a long life. We can remember by the term long life. What are the examples of pro-apoptotic genes? BAC and BAX. And examples of anti-apoptotic genes are BCL126 and BCL-XL and MCL1. Now, the cytochrome from the cell leaks out and combines with AFF1. What is AFF1? AFF1 is apoptosis activating factor. These both, when combined, it is known as apoptosome. What combines AFF1 and cytochrome? When they both combines, they are known as apoptosome. It activates initiator caspase and then executioner caspase. In intrinsic pathway, initiator caspase is 9 and execution caspase is 3 and 6. What does caspase do? Caspase mainly breaks down protein and it has endonuclease activation. Now the extrinsic pathway. Extrinsic pathway is also known as fast ligand pathway. Killing of self-reactive lymphocytes by T regulatory cells. It activates fast ligand. What is fast ligand? Fast ligand is tumor necrosis factor. It activates fast on self-reactive lymphocytes. You can see in the diagram, this is T regulatory. On T regulatory gene, there is fast ligand. Fast ligand, tumor necrosis factor. And here there is fast receptor or TNF receptor. It comes in and then there is FAD associated dead domain and it activates caspase. And then it leads to the activation of initiator caspase and executioner caspase. In extrinsic pathway, the initiator caspase is 8 and executioner caspase is 3 and 6. The macrophage engulfs the damaged cell. This process is known as efferocytosis. How does a macrophage identifies the damaged cell? In apoptotic body, there is flipping out of phosphatidylserine, as we can see in the diagram. Initially, these phosphatidylserine are inside, but in damaged cell, the phosphatidylserine comes out. Therefore, macrophage identifies it. So, macrophage engulfs only apoptotic body and not the normal cell. Therefore, there is no inflammation. What is caspases? Caspases is composed of cysteine. What does it does? It cleaves after aspartic acid. Suppose this is the chain of proteins. As aspartic acid comes, it cleaves from there, as we can see in the diagram. It is in family, initiator and executional caspases are there. Initiator caspase in extrinsic pathway is 8 mainly and 10, and intrinsic pathway is 9. Both of them has same executional caspases 3 and 6, common in both pathways. So we will see briefly differentiating factors between necrosis and apoptosis. So necrosis is mainly pathological and apoptosis is mainly physiological, but pathological examples are also there. Necrosis affects tissue and apoptosis affects single cell or organelle. Now the morphological changes. In necrosis, cell size is swollen, and in apoptosis, cell size is shrunken. In necrosis, the outline is damaged, and in apoptosis, the outline is intact. In necrosis, the chromatin karyolysis is seen, and in apoptosis, chromatin condensation is seen. In necrosis, inflammation is present, and in apoptosis, inflammation is absent. Now, the diagnosis of apoptosis. How we will diagnose apoptosis, we will see further. First, by H&E stain. In H&E stain, we can see the cell is shrunken and the nucleus is condensed. So we can say that apoptosis has been occurred. And there is a special stain which is known annexin V. That stains the flipped out phosphatidylserine. So we can say that it is a process of apoptosis, as only in the process of apoptosis phosphatidylserine comes out, or it is flipped out of the membrane. Third, endonuclease accretion. In necrosis, there is no fragmentation, but in apoptosis, there is fragmentation as it is cleaved in 200 base pair multimers, as we can see 200, 400, 600. In electrophoresis, we can see that A, normal cell is intact, it does not go. B, smearing pattern is seen, and in apoptosis, stripe ladder pattern is seen. Fourth, terminal UTP neck end labeling. What is UTP? UTP is uridyl triphosphate. So this technique is used to see whether the DNA is broken or not. Thank you. I hope you understand what I teach. I will try to be better next time. Please, if you have any suggestions, please you comment down, so I will make videos on them. Thank you.